So today our topic is characteristics of enzyme catalysts. So we have discussed about enzymes. These are used as catalysts in biochemical processes. So they have important characteristics like efficiency, specificity, optimum temperature and pH, increasing activity, influence of activators and coenzymes. So what is efficiency? These enzyme catalysts are very efficient. In a small amount can be highly effective in catalyzing a reaction. One molecule of an enzyme catalyst has a tendency to catalyze, has a tendency to transform 1 million molecules of the reactant per minute. These enzyme catalysts, they can increase the speed of reaction from 10 raised to power 8 to 10 raised to power 20 times as compared to the uncatalyzed reaction. Renin एग्जाम्पल हम लेंगे रिनन की एग्जाम्पल रिनन जो है इट इज एन एंजाइम इट इज यूज्ड टू कोगुलेट इट इज यूज्ड टू कैटलाइज कोगुलेशन ऑफ मिल्क into cheese. So this renin has a tendency to catalyze million times its own weight. It has a tendency to coagulate, to catalyze over 1 million times its own weight of milk protein. So now second is specificity. Specificity means these catalysts are specific in nature. Specific in nature. What that means? That means a particular catalyst, a particular enzyme can catalyze a specific reaction, not all the reactions. इसका ये मतलब है so a particular catalyst can catalyze a specific reaction and cannot catalyze more than one reaction, or it cannot catalyze all the reactions. So this is known as specificity. For example, urease is an enzyme. It can catalyze hydrolysis of urea into carbon dioxide plus ammonia, but it cannot catalyze N-methyl urea. N-methyl urea कोई catalyze नहीं करता, और any other amide या किसी दूसरे amide कोई catalyze नहीं करता. So that means these are completely reaction specific. They possess the property of specificity, I mean to say. The third is optimum temperature and pH. So अगर हम लेंगे ह्यूमन बॉडी का टेम्परेचर दैट इज 37 डिग्री सेल्सियस, पीएच होता है ऑलमोस्ट 7.4. जो कैटालिटिक जो ये एंजाइम्स होते हैं दे शो मैक्सिमम एक्टिविटी at a particular temperature. For example, human body का temperature 37 degrees है, so they at this particular temperature they show maximum activity. ह्यूमन बॉडी का पीएच होता है 7.4 या फिजियोलॉजिकल सिस्टम का होता है पीएच 7.4, सो दे शो मैक्सिमम एक्टिविटी एट दिस पर्टिकुलर टेम्परेचर, दिस पर्टिकुलर पीएच. सो द टेम्परेचर एट विच द एंजाइम कैटलिस्ट शो मैक्सिमम एक्टिविटी दैट इज नोन एज ऑप्टिमम टेम्परेचर, and the pH at which enzyme catalysts show maximum activity that is known as optimum pH. Below this temperature, above this temperature, the activity will fall, the activity will decrease. Exactly below this pH or above the pH, above this optimum pH, the activity of the catalyst will become less, that will fall. So, in short, we can say that these enzyme catalysts show maximum activity at a particular temperature and at a particular pH. The particular temperature is known as optimum temperature and the particular pH is known as optimum pH. The fourth property of these enzyme catalysts is their activity gets increased. That means they show increasing activity in presence of activators and coenzymes. What that means? So if some metal ions are present along with the catalyst, for example, we have Na positive ion, Mn2 plus ion, CO2 plus cobalt 2 plus ion. So if these ions are present along with the catalyst, they enhance the activity of the catalytic activity of the enzyme. So these are known as activators. उनको बोलते हैं activators. So इसका ये मतलब है the enzyme जो catalytic activity of the enzyme है that can be enhanced by adding activators. That means some metal ions, Na positive, Mn2 plus, cobalt 2 plus. Also, if some non-protein part like vitamin is present along with the catalyst, क्योंकि प्रोटीन catalyst होते enzymes होते हैं proteins. So अब अगर कभी कोई non-protein part होगा present along with the enzyme, what this non-protein part do? It enhances the activity of the catalyst and is known as, इसको हम बोलते हैं coenzyme. So that means coenzyme is actually non-protein part present along with the enzyme which activates, which enhances the activity of the enzyme. उसको हम बोलते हैं coenzyme. So enzyme catalysts, they possess the property. What is the property? That means their activity gets enhanced in presence of activators and coenzymes. Also कभी क्या होता है, कुछ ऐसे सबस्टेंस होते हैं जिनसे एंजाइम्स की यानी कुछ ऐसे सबस्टेंस प्रेजेंट होंगे वहां पर कैटालाइज्ड रिएक्शन में, जिनसे एंजाइम की जो एक्टिविटी दैट विल गेट इनहिबिटेड, वो डिक्रीज होगी. उनको हम बोलते हैं कैटालिटिक या एंजाइम इनहिबिटर्स बोलते हैं उनको, एंजाइम इनहिबिटर्स, क्योंकि they inhibit the activity of the enzymes. इसी तरह there are two additional terms which we must remember. दो additional terms भी जो हमें जनरल कैटलिसिस में हमें याद रखनी है. कैटलिस्ट में ये दो terms आती हैं. Mostly we use these two terms in the process of catalysis. One is known as catalytic poison. The second is catalytic promoter. These are two terms used in catalysis. So we have some substances arsenic like carbon monoxide. If such substances are present in a particular reaction, in a particular catalyzed reaction, what they do, they decrease the activity of the catalyst. That means they inhibit the activity of the catalyst. So they are known as catalytic poison. उनको हम बोलते हैं catalytic poison. So catalytic poison or the substances, they are the substances present in a catalyzed reaction which lowers the activity of the catalyst. Exactly, there are some substances, for example, in the preparation of ammonia in Haber's process. मतलब Haber's में हम बनाते हैं अमोनिया. यहाँ पर हम करते हैं कैटलिस्ट इस्तेमाल आयरन. सो यहाँ पर आयरन के साथ हम करते हैं मोलिब्डेनम भी यूज. व्हाट दिस मोलिब्डेनम डू हियर? दिस मोलिब्डेनम एक्चुअली इट प्रमोट्स the catalytic activity of this iron, that it enhances the catalytic activity of this iron. So it is known as, this molecule is known as here catalytic promoter. So what is catalytic promoter? Catalytic promoter is a substance which is present along with the catalyst and promotes its activity, और उसकी वो activity promote करता है, catalytic activity करता है promote. So that is said to be catalytic promoter. So this was about characteristics of enzyme catalysts, and we have at last we have discussed two additional terms here: catalytic poison and catalytic promoter. Thanks.